Good to see everyone back here for day 12. Today yesterday we talked about quality control, loading in expression data, genomics data, the structure of it, the format of it, some of the public databases that we can use. I wanted to start today by talking about the lab from yesterday. The solutions are available on the GitHub site, and the best way, for you to get the solutions yourself is probably to download the .qmd file and save that in your files in the week 3 day 11 folder. I'll put the link to this in the chat. So if you download this solutions QMD file right here, click on it, you hit this little download button, and you save it into your folder, the week 3 11 folder right here. If you do that, then you can open it up in our studio and render it yourself, and everything should end up looking like I have right here. So the lab was working on the Pasilla database, so a little different one. Some of the differences in it was that this was data that did not come in that summarized experiment format. The counts and the metadata were separate. So we first downloaded them and then created the summarized experiment object. Let me close my door real quick. And so you can see what the assay data looks like, the dimensions of it, the conditions per sample, 3 and 4. So it's slightly unbalanced, but not severely so. The library type balance, again, so among the treated, we don't have the same amount of pair end and single read, but we couldn't because there's 3, and that's an odd number. But so again, yeah, relatively balanced, but not completely balanced. So the next part was looking at the count distribution and low count filtering. So here is the first sample, the raw counts, only non-zeros in the log 2 count +1. I see those looked very similar to what we had in the airway database. 2,200 genes with 0 total counts, which is 15.3%. If we filtered low count genes, defining as those with a count less than 10, we would keep around 10,000, drop around 4,500 of the genes that we started with. And this is a good thing to do, and we'll do this again when we move forward. Helps out from a couple of standpoints of what does this filtering do. This is the density plot of those log 2 counts for the 7 different samples, and these should look relatively similar. There should be some good overlap between those, which we see here. There's some kind of natural ordering here, kind of trickling from one to the next, to the next, to the next. That's what we like to see. None of these are outlying to a great degree. The next part was looking at just some quality control measures. So here we have the PCA results. So we have principal component 1 on the x-axis, principal component 2 on the y-axis. So number 1, we see a clear difference between treated and untreated by principal component 1. So those with positive scores are treated, those with negative scores are untreated. So that's a good sign that the treatment had some notable effects on the expression levels that we got, right? The second thing that we see here is the library type. There's definitely a clear difference in the pair-end and the single read library type in terms of principal component 2. So those with positive scores on principal component 2 are all single read, and those with negative scores are all paired read. So that is a little concerning. So in situations when we have technical variables that are showing a clear impact on the results that we get, that's something that's going to make us a little concerned, right? Because here we don't want library type to be driving the results. We want condition to be driving the results. Now, the saving grace here is that condition is sort of the main separator here when we look at PCA1, which is the PCA that accounts for most of the variance in the data. So that's a little bit of a saving grace, but a little concerning that we see library type having such a big influence here. So we'll look at running this adjusting for library type when we look at this data set again. And we'll do that because it seems like there maybe is some impact of library type. Something else I wanted to show you. Sorry. So I'm just pulling up the lab file, just this library type issue. This is something that we noticed at this point when we were just looking at the library sizes. So the 3 with the biggest library sizes are treated 1, untreated 2, untreated 1, and untreated 2. And if we look back, treated 1 is single read, untreated 1 and untreated 2 are single read. So the library sizes are higher in the single reads than in the double read. And the PCA analysis we're doing right here is it may be picking up some of that, some of the library size artifacts, but this is something we'll look at adjusting for towards the end of the morning section here, and in the lab, that's something that we'll look at as well. Any questions about that? Okay. All right. Okay, well, let's talk about our main topic for today. So the slides are available in the week 12 folder. The file did not have .pdf on it, so if you try to open it and you get an error, like your computer doesn't know how to open it, you could either try adding .pdf, or if you try to open it and it asks you, what should I open this with? Just choose Adobe or Preview, however you view your PDFs on your computer. So that was a mistake I made when naming the file. Okay, so today we're going to talk about DESeq2, which is a suite of tools applied with a single function in the DESeq2 package. This DESeq2 is going to use those summarized experiment objects that we were talking about yesterday, count matrices and column data mainly. And it aims to answer the question, which genes change their expression levels between treated and untreated cells. This is called a differential expression analysis. So in the airway data set, we have human airway smooth muscle cells treated with dexamethasone or untreated controls, 8 samples across 4 cell lines. As we mentioned yesterday, there's about 20,000 genes we're going to be looking at here because there is a filtering, so it's not going to be all 60,000. It's about 20,000 we end up testing, and what we want to test is the null hypothesis. The mean expression is the same in the treated and untreated cells. So it creates 20,000 separate hypothesis tests, 20,000 separate p-values. So some of the challenges that come up in this, with 20,000 tests, if we just rejected everyone at a 0.05 level, we know that there would be about 1,000 false positives. So way too many false positives. So we have to adjust the p-values after we do the analysis. 2, there's small sample sizes. So in the airways data set, we only have 8 subjects. And a lot of these studies will only have, say, 3 to 6 samples per group. So variance estimation from only 3 replicates or 6 replicates is going to be very unreliable. This is something that in standard statistics class, you would never do an analysis with only 6 samples or 8 samples or 7. So like it's just, completely, it's not done. But here, this is what we want to do. We want to make use of this data. We want to see what it tells us. The DESeq2 function that we use is going to address both of these issues, stabilizing the variance estimates across all the analysis that we run, and use something called the Benjamini-Hochberg procedure to control the false discovery rate. So let's go to the file for today. Make this a little larger. Load in all the packages that we need. So here we'll just use the airway data set that we used last time. And this is one of the summarized experiment objects. Has everything in it, has the count data, has the column data, has the row data, everything we need. So when we run this, usually when you have 2 groups, treated and control, the standard method in statistics is a 2-sample t-test. That is used for continuous data that is either approximately normal or has a large sample size. And as we know, this data, neither of those are true. It's not approximately normal, doesn't have a large sample size. Also, some t-tests, the variance will be assumed the same for both groups. That's also not going to be the same. So RNA-seq data that we're looking at, those counts are going to violate both of those assumptions. The counts are non-negative integers, so they're not continuous. There's a lot of counts at low values, so they're very right skewed, and the variance grows with the mean, which is something we'll look at in the data that we have. So one of the common models to use for RNA-seq counts is this negative binomial distribution. So the negative binomial distribution is for count data, and the variance of the negative binomial distribution is equal to the mean plus alpha times the mean squared. So the variance is a quadratic function of the mean, how strong that quadratic curve is controlled by this alpha dispersion parameter. If alpha is equal to 0, then it's not quadratic. It's just linear in the mean, which is the same as the Poisson distribution. So RNA-seq data almost always have this alpha being greater than 0, so has more variance than the Poisson. And let's take a look at this. So why don't we use a t-test? Well, first off, we have 4 treated and 4 untreated. So, we can't assume that the central limit theorem is not going to apply here. So that's one reason. The second reason, when we look at the count distribution here, it's not anywhere close to normal. It's very, very right skewed here. So the other thing is the variance in the overdispersion, and that's what we see in this right figure. So what this right figure shows, it's not looking at every gene here. We're using this slice sample function to randomly select 3,000 genes out of 33,000. So we're looking at about 10% of the data just to demonstrate the relationship between the mean and the variance. And those are these dots right here. So for 3,000 randomly selected genes, these are the means and the variances, both on a log base 10 scale. So the red line here, this is where the mean would be equal to the variance. That is not the case here, particularly for those when the mean is large. There's clearly a much larger variance than there is a mean. So what this shows is that the variance grows with the mean, and it grows even faster than the mean does. So this is an indication that the negative binomial model that we are employing is going to be fitting to use this. So again, the negative binomial model, the variance is quadratic, okay, with the mean and the mean squared. So there should be a quadratic relationship, some type of quadratic relationship with these two. Yeah, so DESeq2, what it does is it estimates this dispersion parameter, this alpha parameter, for every gene. And then given that dispersion parameter, it tests for differential expression between the treated and untreated using that negative binomial model. We're only going to have 3 to 6 replicates per gene. So with that few number of data points per gene, estimating the dispersion from the data is going to be very noisy. There's going to be a lot of noise in it. When you do something 33,000 times, just weird stuff is going to happen. For example, when we look at the figure down here, there are some data points where the mean is less than the variance. Okay, so we see some blue data points down here when the mean is lower, that the mean is actually less than the variance. That's probably just random variation. So weird things happen when you do something 33,000 times. If you flip a coin 8 times and you repeat that 33,000 times, you're going to get some weird things. You're going to get some all heads, you're going to get some all tails, things like that. So weird things are going to happen. So when we estimate the dispersion parameter for each gene separately, we're going to get some estimates that are wonky. And we want to control for that. And so DNAseq2 is going to do this for us. So what it does, it's something called empirical Bayes shrinking. We're not going to talk about the details of it, but it takes the dispersion parameters for all genes in our data set, and it looks for a trend between the dispersion parameter and the mean. So between the dispersion parameter and the mean of the normalized counts. And it uses that trend to sort of correct the dispersion parameter, the gene level dispersion parameter estimates, just to make them a little bit more reliable than they are when we just look at each gene separately. We can plot this using this plot dispersion estimates function. We have this figure right here, but I wanted to look at this in a little more detail. So I'm going to pull up this day 12 differential expression .qmd file, and I'm going to run this into our studio, and so we can look at this. So I'm going to just run these code chunks one by one. I don't need to run all these, but we can run them all. For example, if we wanted to see P2 a little closer, we could look at that. Okay, this is a little closer look at that mean versus variance relationship. Interesting. And I think there's I'm gonna have to kind of skipped a couple things here. So I think I have this a little out of order. Sorry about that. I have some filtering stuff in here. But I want to get down to this, this plot. Here it is. So this is the plot that shows us the dispersion estimates for all genes that made it into the filtering, that made it past the filtering. So there's a few things that that we see from this. The red line, this red line represents the trend between the means, the normalized means. So how many reads per gene there were, and the dispersion parameter estimate. A higher dispersion parameter estimate means that, so that's that alpha parameter. So mu plus alpha mu squared. So this is the trend. The dispersion parameters tend to decrease as the mean increases. So there's a slight decreasing trend, and then it levels off towards the end here. So that is the trend that we see between these two values, and that's just done with a flexible regression model using the splines that we talked about in week 2. The black dots are the actual gene-specific estimates of the means and the dispersion parameters. So you see all these black lines down at the bottom, so that kind of bunched down in here with dispersion parameters equals 1 times 10 to the -8. So that's a 0.8 zeros and then some value after that. So this is the smallest that these dispersion parameter estimates can go. So what happens is if a particular gene has a variance that's less than the mean, then this dispersion parameter, it's always going to be better and better and better and better and better the closer and closer you get to 0. So what this is saying down here, all these genes here, these are situations where it's likely that the mean is actually larger than the variance is, which, hey, that can happen. We're doing this 33,000 times. There's going to be some situations that can happen. Majority of the time, it does not happen. So those are those low estimates, but most of the black estimates you can see are up here in this, pretty close to this red line, with a few down here below, a few down here above. Okay, so what happens is we take the trend line and we take these black estimates, and the black estimates get brought up towards the trend line. They get shrunk towards the trend line, and this is this empirical Bayes shrinkage procedure that we have. So we assume that around each mean, so around each red point, there's some distribution of these dispersion parameters, and we take the value we observed, that distribution around the mean line, and those get put together for our final blue estimate. So all these points down here, they all get shrunk towards the mean. They all get brought to a larger value, and this is going to help stabilize the variance estimation. This is acknowledging the fact that we have very few replicates per gene and saying, well, these black dots we're getting are not going to be reliable. Let's try to use the collective wisdom of all these black dots to try to get better estimates. One thing you may notice is that, while we shrink all these low estimates up, right fairly close to the fitted line, we don't quite do the same for the estimates that are very high. So these points up here, they barely get shrunk at all. And in fact, they look to be almost the same thing. So when the dispersion estimate is greater than the trend line suggests, the shrinkage is not as great as when the dispersion estimate is less than the trend line. So why is that? Well, I can speculate here, putting my speculative hat on. So I believe this is because the p-values are going to be smaller the smaller this dispersion parameter estimate is. So when we get a small dispersion parameter estimate, that's more likely to give us a significant p-value. When we get a large dispersion parameter estimate, that's less likely to give us a significant p-value. So here we want to be conservative. We don't want to find significance in one of these genes just because we got a wacky dispersion parameter estimate. So that's why we're putting it way up, and we're saying, okay, let's let the means of the treated and untreated drive the p-values more so than these dispersion parameter estimates we're getting for each gene. These ones up here, we're not as concerned about these giving us significance, because when you have higher dispersion, you are going to more likely have non-significance. It's going to inflate the p-value more than it's going to deflate the p-value. I don't know that for sure, but that's kind of my statistical thought about why this would be the case. So I think this is very interesting. I was kind of learning about this as I was going along, but I think it's a really cool thing that they do to sort of, hey, let's pool all the information together to try to get better estimates on a gene to gene basis in a smart way that is going to help our results be more reliable and robust. Any questions about that? So this is something to take a look at. I think this is more for your conceptual understanding, just more for your knowledge about what is going on behind the scenes when we're running this type of analysis. I don't think you would. I'm not positive how you would use this in practice, how much impact this would have on your analysis. To me, I think it was just more of interest conceptually, what's going on here? How is this model working? So, all right. Okay, so the next part of this is normalizing for sequencing depth. So this gets to the point that the total number of reads that we have is going to be different sample to sample. How DESeq2 controls for this is it estimates a size factor for each sample, which is going to be one number that captures how deeply that sample was sequenced relative to others. How it estimates this size sample is not, it's not particularly meaningful to us, but what it does is essentially it uses the total number of reads per gene and does some summary statistics on that, and we get a size factor that spits out of there. And then with that size factor, we get the normalized counts, which is going to be the raw counts that we get from our matrix divided by the size factor that is calculated in this step. We don't do this manually. DESeq2 does it all internally. We will look at the size factors for this analysis we're going to do. Okay, so let's look at the full workflow here for DESeq2. First step is we are going to build the data set. So provide the counts, sample metadata, and the design formula. Second, we are going to, DESeq2 is going to filter the low count genes. So that removes genes with very few total reads across all samples. These are going to carry little information and slow the computation of the function. Set the reference level, so tell DESeq2 which group is control so that fold changes are in the direction that we're expecting. And usually the fold changes have the treated on the numerator, untreated in the denominator. We run the DESeq function, one function call. It's going to estimate the size factors, estimate the dispersion. It's going to correct the dispersions using that pooling process we talked about. It's going to give us p-values, adjusted p-values, so on and so forth. And from there, we get the results, which are going to give us the average normalized count across all samples. This result is going to have this information for each gene. The log2 fold change, which is log base 2 of treated over untreated. The standard error of that log2 fold change. The Wald statistic, that's going to be our test statistic, kind of like our T statistic. This one has a Z distribution. We have the raw p-value and the adjusted p-value that's adjusted for multiple comparisons. So let's take a look at how this looks for the data we're going to have. First thing we want to do is we want to verify the alignment. We want to make sure the sample metadata are aligned with the rest of the data. And here, the counts and the sample metadata are aligned. We should also ensure that the rows are aligned with the row information. That's another point that we want to do. The next step is building the DESeq data set. So to build this, we create this object DDS. So that's our DESeq data set. Using this DESeq data set from matrix function, we give it the counts. And the counts we created way up above here. Just using the assay function. That's what gives us the matrix of counts. We give it the sample info. This is the column data that was created right here. So just did col data airway. I did as.data frame just to give it a little better formatting. And then the design matrix. So here we are telling this is where we tell the DESeq function how to model this data. So DEX, that is a variable in the sample info that tells us which of the samples are untreated, which of the samples are treated. We can add other things to this design matrix, which we will do later on. We will add the cell type to see what kind of impact that has. And then we just printing this out says, okay, you created a DESeq data set, has all the counts in here, all 63,000, and count matrix, row names. These will be the encoded names of the genes. Column names are the ones in the sample. And then here's some other information about the col data that we gave it. The next step is going to be the filtering. So here we are going to filter those with at least 10 counts when we sum across all samples. So we have 8 samples. We're going to sum across all 8 of those samples, and we need to have at least 10 counts. So that gene had to come up at least 10 times among these 8 samples that we have in our data. So why do we do this? Well, it makes things faster, so the computational time goes down, and it decreases. So including these samples would waste computational time, and it would slightly increase the multiple testing burden. So this DESeq function, it adjusts for multiple comparisons. It adjusts for the fact that we are doing a lot of tests. The more tests we do, the more we have to adjust. So by limiting the data set, we're making that multiple testing burden, we're decreasing it. So instead of doing 63,000 tests, we're doing 22,000. Still a heck of a lot of tests, right? But it decreases it to some degree. The downside of doing this is that there are 41,000 genes that we did not test. So there are, when we ran this experiment, we said, oh, we got 63,000 genes that we're measuring. We actually are only testing 22,000. The other 41,000 we're throwing away. But we had so few reads in those 41,000 that we really couldn't test them. If we would do this again, we would want to have more reads. And as I think May mentioned yesterday, if we had more reads, we can get some of those reads from some of the rare genes. So that's an indication that these, we just didn't get anything there, so we're removing them there. Next, we're going to set the reference level. So this DEX is our treatment, and I'm going to use this rel level function where I'm going to set the reference level as untreated. So going back to our results, the log2 fold change, we want that to be treated over untreated. So on the denominator is going to be the reference level. So I'm setting the reference level. excuse me, setting the reference level to untreated. So that tells this DESeq2 function that, hey, let's have untreated on the denominator. Sorry about that. Too much talking. So please, if you have any questions, save my throat and ask them. Okay, so the next part of this is running the entire workflow using the DESeq function. I did this earlier while we were running this through, and you can see it gives us sort of a blow by blow of what's happening. First, estimating the size factors, and this is what allows us to normalize. Estimating dispersion, gene-wise dispersion estimates. So then we're doing the mean dispersion relationship right here. That gives us the final dispersion estimates. And then we fit the model and we test. These beginning steps are usually what takes most of the time, but it does not take this runs relatively quickly. I mean, so, for doing all that, what did that take? Five seconds or so? It's a pretty efficient function. We can look at the size factors. So this is how the reads will get normalized by these size factors that DESeq calculates. So the largest size factor here is about 1.18, or no, 1.4, excuse me. Smallest size factor is about 0.7. So not too large. If we saw something that had a size factor of 5, then it's kind of like, all right, well, have some library that's outlying here. So it's another look at the look at those library reads and just make sure they're all in a relatively similar range. But here everything looks good. Dispersion plot we looked at. And then here we're extracting the results, looking at using the results function, and you can see the first 5 or 6 columns here of the results. So we have the base mean, 708 for this first gene, 520 for the second gene, the log2 fold change, the standard error of that. This is the Wald statistic, the Z statistic that gives us the p-value. And then finally, we have the adjusted p-value. So let's go through these. So the log2 fold change, this is going to tell us the direction and magnitude of the expression change. Some of the reference points here. If the log fold change is around 0, that means there's no change in expression. So if it's 0, that means the mean of the treated was the same as the mean of the untreated, because log2 of 1 is equal to 0. If the log2 fold change is 1, that means that the treated cells have twice the expression as the untreated cells, because 2 to the 1 is equal to 2. Log fold change is -1. That means treated cells have half the expression of the untreated cells, because 2 to the -1 is equal to 0.5. In practice, the 1 and -1 are kind of used as goalposts. So anything we find with log fold change less than that is kind of disregarded to some extent. It's like we want to see log fold change that are at least twice twice as show double the difference between treated and untreated in one way or the other. So anything 1 or greater or -1 or less, this is going to give us some things that we're not quite as interested in. If there is very low mean expression, the log fold change estimates can be a little variable. And why is that? Well, when you look at the ratio of two random variables, so here we're looking at the ratio of treated to untreated. When that denominator is small, there's just a lot more variability to it. Small changes in the denominator. If this denominator, say, is 0.001, this whole log 2 fold change is going to be a lot more sensitive to just small changes in that denominator part. So when we see very low mean expression levels, even if the LFC is large, we kind of want to discount it somewhat. And this is why we account for that. We look at that p-value. The p-value is gonna account for that for us. So we're going to take both of these things when we make our final assessment of, hey, what variables should we really be focusing on? We're going to use the adjusted p-values, and we're going to use the log 2 fold change estimates. Those adjusted p-values, just a quick slide on those. We'll talk about these more in depth in day 13. But the main idea here is that if we cut off the p-values at 0.05, we would expect a whole bunch of false positives. So the alpha 0.05 is not really going to be that meaningful. The false discovery rate, what this is going to do is control the expected proportion of false positives. So out of all the significance results we get, how many of those do we think are going to be false positive? What proportion are going to be false positives? And the particular procedure is called the Benjamini-Hochberg procedure to control the number of to control the false discovery rate. So this p-adjust has been adjusted for multiple comparisons using this Benjamini-Hochberg procedure. Some of you may have heard the Bonferroni procedure before. Bonferroni is another multiple testing procedure. It's one that requires a lot higher threshold for saying something is significant versus the BH procedure. So we'll discuss that more in depth when we get to day 13. So we have the p-values here, and we have the adjusted p-values. So this first one is a good example. The regular p-value is less than 0.05 at 0.0286. So it's going to be less than that. The adjusted p-value is not. So the adjusted p-value is always going to be higher. And here it knocks that one from being significant to being not significant. When we do the summary of the results, it says out of the 22,369 with non-zero total read, if we look at those that have an adjusted p-value less than 0.1, commonly we'll use 0.05, but here it uses 0.1. We see how many of those have a log 2 fold change that is positive. So 1,884, about 8.4% of the genes we look at, the treated has a higher expression level than the untreated. For those that are less than 0, there's 6.7%. So 6.7% of those that have that adjusted p-value less than 0.01, 6.7% of the genes are going to be less than 0. Let me rephrase that. So out of all the genes, 6.7% have an adjusted p-value less than 0.1 and are down. The untreated has a higher expression level than the treated. So there are more on the positive side than on the negative side, but there's a lot of each. It tells us how many outliers there were, 51 of those. And you can see that uses this Cook's cutoff to do that. Low counts. So there's another around 4,000 that were removed. So we removed about 40,000. This took another 4,000 away because the mean count was less than 4. So this particular filtering here, this is going to use the normalized counts. When we initially did our filtering, we did not do the normalized counts. We used the unnormalized counts. So a few more are taken away by the DESeq procedure. Okay, so here we're going to look at some of the top differentially expressed genes. So we're taking the results. I'm going to go to R and just try to catch up with some of this stuff. Okay, so we have our results here, summary of those. And here, I'm just changing the results into a data frame. And I'm taking the row names. So this res has row names to it. And the tables that we use, they don't like row names. So it's one of the 10 commandments of how tables work is you shouldn't have row names. So we're using this row names to column function where this will take the row names from res and turn it into a column gene ID that will have that will contain the row names. So now this new data frame that we've created, no row names to it. They've been stuck over to this gene ID variable. And then we are turning it into a tibble, which has some nice properties to it. And then finally, we are arranging by the p-adjust variable to so that the genes with the smallest p-values are elevated to the top. So these are the most significant genes that we have. So here we see this is the gene ID for the most significant genes. It's just out of curiosity, we can look at let's look at the data for this one. Why not? So I'm going to say counts. Counts does have row names. And I want to look at the row where the row name is equal to this code, which is just what we get up here. So we see the most significant one has a base mean of about 1,000. The log2 fold change is 4.6. And we can see the data here. So the log2 fold change is 4.6. We can take 2 to that power. And that means the mean of the treated is 24 times larger than the mean of the untreated. So huge difference between these two. Okay, so here's the data. Of course, we need to know who is treated and who is untreated here. I think it goes. So let's look. I think it's sample info. Okay, yeah. So it goes untreated, treated, untreated, treated, untreated, treated, untreated, treated. So we have untreated, count of 62, treated, count of 2,040, untreated, 99, treated, 1,100, untreated 99, treated about 2,000, untreated 79, treated 2,000. So a clear difference between the treated and untreated, obviously. But so that's one of the main reasons why this gene goes all the way to the top. The other reason is the dispersion of this particular gene, I would guess. So you may notice that not only the untreated are lower, but they're actually relatively close to one another. So there's not a lot of difference between those. And the treated, three of them are right around 2,000. We have this one sample, which is lower. These aren't including the normalizing factors. So probably a reason why this variable jumps up as much as it does is a combination of those two things. The data for the treated and the data for the untreated are relatively similar. There's not a lot of variance, so that's going to drive that dispersion parameter lower. It's going to make things more significant. That's one part of it. The other part of it is the treated have a mean that's 24 times the untreated. So there's a clear difference between these two. And with that, we get a p-value that 9.25 times 10 to the negative 105th. And even when we adjust it, it's times 10 to the negative 100. So 0.100 zeros and then some values after that. I don't even know how you calculate values that small. Most computers, they kind of fall apart when you get to these tiny, tiny numbers. Yeah, so that was fun. Let's look at the second most significant one. So the second most significant one, the log2 fold change is not nearly as large, only 1.45. So it doesn't seem to be as much on the mean side. If we take 2 to the 1.45, so the treated have 2.7 times the mean that the untreated do. Does appear to be a clear difference between these two. There is definitely a difference. The means here are relatively high. That probably helps out the significance of that. We can see that the log2 fold change standard error estimate is very low in this situation. That's probably driven by the fact that this base mean is a little bit higher, but also by the dispersion parameter. So all these things are combining to give us these p-values that we get. Any questions or anything you want me to look at on this? Okay. Yeah, I wish it gave us the dispersion parameter estimates as part of this results part. I'm not sure if we can. I can look at question mark results to see. Sometimes you'll see here like there'll be an option that will allow us to get some more data with what we get back. Some good details here. Yeah, I don't see anything about that dispersion parameter estimate. For another day. Okay, so here we're looking at those that are the most significant. In this one right here, we're going to use the row data to try to add some information to our results. So the row data has information on the genes that are in the airway data set. All I'm going to take here is the gene ID and the gene name. The gene ID is what we are going to merge these by. So using this left join, this is what we talked about in week 1. So I'm taking the results and I'm joining the gene information to those results by gene ID. So I'm just merging them. So in this situation, even if the ordering of the results is different than the ordering of the gene info, we're okay. And in fact, it is different because we've sorted these results by the adjusted p-values. So the ordering is not going to be the same. That's all right because we're using a left join here by gene ID. So those are going to match. It is going to do the reordering for us when we do this. And now I'm just selecting a few of the variables. Same thing that we had before, just now we have the gene name on here. We could add some more information. I think I already ran this, but let's run that again. So I could not just use the gene ID and gene name, but take all the gene info and I'm just going to run these 2 lines. So, and now when I take a look at this, we have a little more information here. The start, the end, sequence name, sequence strand. I mean, the gene biotype, that's something that I think we might want to hold on to. So let's add that up here. And then I'm going to add that to my results down here. So now we have the gene biotype with our results as well. And then with the gene name, we could look up some information on that. Any questions about what we got so far? Okay. I think we're at a good space to stop here. We're at 11:03. Let's take a quick break and come back at quarter after at 11:15, and we'll continue on with exploring the results. Okay, well, let's continue on from where we were, and please add to the chat or unmute yourself and let me know at any time of any questions that you have so far. The last thing we looked at was we just looked at the top genes. Again, this time we just added the gene name to it. And when we looked at this in R, we also added the gene biotype if we wanted to have that information. How many significant genes at the 0.05 level? So here we're adjusting, we're filtering by p-adjust less than 0.05, and we're looking at how many there are total, how many are upregulated, how many are downregulated, and we can see the results right here. More upregulated than downregulated, but a good amount of both. I haven't done a lot of these analysis, but this certainly is a lot of significant genes here. 2,700 or so. More than we could probably investigate one by one, certainly. So maybe, hey, let's just look at some of the top hits here. I'm going to copy this and paste it over into RStudio in a second. So here we're looking at the top 5 upregulated and the top 5 downregulated genes. So we're starting with the results, filtering by 0.05. And then we're using the slice max function for the log to fold change and only getting 5 of those. So what this will result in, this slice max function will only keep the 5 rows that have the largest log to fold change. We're doing the same thing down here, taking the results and filtering by 0.5. This time we're using the slice min function. So we're only keeping those with the smallest or largest negative log to fold change. And then we're using bind rows to just stack these on top of one another. Only keeping a little bit of information here and creating some other variables. The log to fold change, just rounding it. P adjust, just making sure it gives us it in the way we want it to have with that E. So it's times 10 to the whatever value we give it. And this is the data set that we run. So I may want to include some more information than this so we can look. So I'm going to delete this select line. So I'd like to have a little more information. Okay, so then we can see the gene that has the largest log2 fold change. So that log2 fold change is 9.6. So the ratio of the treated to untreated, we would do 2 to the 9.61, 781. So the mean of the treated, 781 times the mean of the untreated. Wow, that's a huge, huge difference. On the other side, we get negative 4.36 to the negative 4.36, 0.04. So the mean of the treated is 0.04 times the mean of the untreated. So the mean of the untreated is quite a bit larger in that situation. But just out of curiosity, I would like to look at the data for this this gene, so we can use that counts data again, and the row names of counts equal to, quote, this gene ID. Boom. Okay, so this is the gene that has the biggest difference between the treated and untreated. Yeah, well, very interestingly, all the untreateds are 0. So we don't have any non-zero counts for the untreated. So the mean there is going to be very, very small, obviously. And then the treated, we have some counts here, not terribly high counts, but certainly there's a stark difference between those two. So one important thing that this points out is this log2 fold change is more than simply the mean of the treated over the mean of the untreated, because that would not be defined in this case, because the mean of the untreated here would be equal to 0. There's more that goes into it than that. Let's take a look at this second gene that we have. This one has a larger base mean. Wow, yeah, clear difference there. Very large difference between the treated and untreated in this case. Log2 fold change 7.18. So these are all things that we might want to take into account when we are considering, hey, if there's, 10 to 30 genes that we can really look at, the p-adjust is important because it does give us significance, but also the log2 fold change is important. And we probably want to, have some, take both of these things into account when we're figuring out which variables we may want to look at. And let's take a look at some of the down-regulated ones, I guess, just to be complete. Okay, so here's a situation where the untreated have a larger mean than the treated. low counts here again. So here the numerator, the treated have only 2 non-zero reads to it. So that's an indication this base mean is very low. And it's probably why the significance isn't quite as high in this situation. So interesting way to look at this data and see what some of the important results were. Any questions on any of the results? Anything you want to look at? Okay, so here we're going to look at some figures. First, we have the MA plot. This plots M, which is the log2 fold change on the y-axis, A, mean expression on the x-axis. Each point in this figure is one gene. Significant genes, so those that have a p-adjust less than 0.05 are highlighted. So those are going to be the red points. So what do we look for here? Most genes are around this midline where we have the log2 fold change close to 0. I'm gonna plot this here so we can look at this figure in a little more detail. So most genes are going to be around this midline point where the log2 fold change is around equal to 0, indicating that the treated and untreated have similar counts. For the low expression genes, so when the mean of the normalized counts is lower, notice there's wider spread in the log2 fold change. So this is an indication that when the mean is lower, the log2 fold change has more variability to it. The p-values that we look at, they take this into account. So notice that where the gray points are for means that are down on this end is a lot wider than where they are up on this end. So up here, red points occur for very small log2 fold change, like around 1/2. You're already significant. Down here, when the mean is lower, even when you're out past -4 or +4, sometimes we still don't have significance. So this kind of outlines the importance of the adjusted p-values, the p-values and the adjusted p-values. They are going to filter out a lot of these situations where we get very large log2 fold changes, but they don't really matter because, there's not really a lot of significance there. It's just that when we're down in this lower region, things are a lot more variable. When we're up in this higher region, we don't get as much variance in our results. Okay, so we don't want to see a lot of significance down in this region either. That would be an indication that our filtering is not working as well as it might be. Here we've cut this plot off at -5 and 5. We go back to the code. See, I set the y limit equal to -5 and 5. So we don't have any values that are outside of that. There are values that are larger than +5 and smaller than -5. Actually, I don't know if there are any smaller than -5, but these triangles that we see, that's just kind of an indication that the true point is actually outside of what we're looking at. So if we wanted to make this bigger, okay, I don't think we need to go down to -8. Let's just do -6, and then we could bring it up to +8. This will, we'll see some of these points that were actually outside of the first limits that we had. Here's that largest one that we looked at earlier. The dotted lines are the 1 and -1 points, and usually we don't want to look anything that's inside the 1 and -1. So up here, that's an indication that the treated mean was at least twice the untreated mean. Here, the treated mean was at least half of the untreated mean. So we kind of want to use those as sort of goalposts. So what would be of interest are those that are in red, okay? They're significant, and they are outside this 1 to -1 goalpost that we have. So that's the MA plot and what we can use it for. Any questions? Next one is a volcano plot. This one's similar to the last one, but this is going to plot the effect size on the x-axis and the significance on the y-axis. The ones we're interested in here are those at the corners. Those will be ones that are highly significant and have a large log2 fold change. Sometimes you may hear me call that an effect size. The log2 fold change is sort of the size of the effect. And we want to, as I said, we want to be looking at both of these things. Genes near the bottom are going to be those that have low significance. Genes in the middle are going to have small effect sizes here. This is kind of just exploratory for identifying variables that we want to follow up with. Let's take a look at that here. We can look at it nice and big. Yeah, and the ones. Okay. And this is the first gene that we looked at the data of. Log2 fold change is about 4. P-value, adjusted p-value is times 10 to the -100, some ridiculously small value. Here's the other one we inspected. This is the one with the largest log2 fold change, but we can see some others that would certainly be of interest. And some of those down here. So kind of, again, we would want to take both of these things into account, not just significance, not just effect size. If we want to see things that are really promising, we want to combine both of these two quantities. So look at the significance, but then give a little more weight to the effect size here. So for example, this gene down here, it's not, does not have the most significant. There's a lot of tests that have, for example, this one right here is more significant than that, but the log2 fold change was much larger here. So that one will be more of interest than this one over here, for example. And then we see all the non-significant, all the gray down here. Yeah, just one way to look at our results. Okay, next thing we can look at is a heat map of the top differentially expressed genes. This is going to use some clustering that we will talk about tomorrow. The heat map that we have, the rows are going to be the genes, the colors are going to be the samples. The colors that'll be on here will be an indication of the expression level. So we can see some relationship between the top genes and their expression level and how they are related to the treated and untreated. Let's take a look at that. I'm actually just going to go right to R because I enjoy looking at these plots in full. So I'm using the P heat map library here, so you'll have to install that if you haven't done so already. Okay, so this is the heat map. So on the top, we have the columns are the different subjects in the data set. The rows are the different genes. I'm going to bring this in a little bit. It's almost like this is, okay, it's a little better. So the rows are the different genes. There's two lines of colors here. The top color, this is going to be the cell that the sample came from. The bottom color is going to be treated and untreated by the dexamethasone. We have the cell name on the right-hand side, and then some grouping of these cells down the left. So the cells have kind of been grouped by how their pattern of expression levels. The cells we see here on the top, the expression levels for the untreated are larger than the expression levels of the treated. We can tell that because the colors here are red, so reds are a little bit larger than when the colors are in blue. All the rest of the genes have the opposite. So the treatment here, we have larger means than the untreated. We can try to look at some of the cell relationships here. It is a little bit challenging, though, to try to compare the similar cells with how they look to one another. But you can see those cell markers at the top there. Yeah, so clear block pattern here, which is nice. I think that's something that we would want to see. So there's a strong treatment effect among these top 30 genes that we identified.
Hey, Dr. McLean, we had a question about what the brackets mean on the heat map.
Sure. So we see these brackets up here and these brackets down here, and then we have similar brackets on down the rows here. So this is a clustering procedure called hierarchical clustering, where it takes the whole data set and then splits it into two groups, for example, and then we'll split it again and then split it again and split it again until everyone is by themselves. So what this shows is that when we look at the top differentially expressed genes, what I would take from this, the first split splits all the samples into treated and untreated, which obviously would happen because these tests look at the difference between treated and untreated, and they, of course, are going to pick that out. That's why these were the top 30 differentially expressed genes. So that's really what I would take from this top one, that, okay, it just split it into treated and untreated. I don't think we can take too much from, okay, these two were the last two that were split apart. These two were the last two that were split apart. I don't think we can take too much from that. And the same thing going down the rows. So the first split here splits it into those that were downregulated versus those that were upregulated. And obviously, I think that's what we would expect here from the results. We could dig down into these a little bit more, maybe, to get, okay, these two genes right here, these two were both highly significant, and their results tended to be very similar. And we know that because one of the last splits was to split these two apart. So these two were, the longer two rows or two columns are together, the more similar they are. We could say the same for these three. So that can tell us something about, wow, maybe these two genes are very similar. They have different mechanisms. Maybe this is something that is well known in the literature that we already know, and then maybe that tells us, all right, we only need to look at one of these two genes. They're both similar, and they both have some similar mechanisms to them. So this is something we will talk about tomorrow, just in general, clustering and how these procedures work. Yeah, thanks for the question, Chad. Any other questions? Okay, so the last thing that we are going to go over is adjusting for additional covariates in the DESeq2 results. So this first model we ran looked at treatment. It really looked at what is the effect of treatment in the expression levels of the genes. We can add more information here, so it adjusts for more things. For example, the airway experiment, there's 4 different cell lines that were used. If we do not account for cell line, the biological differences between cell lines could inflate the residual variance, could reduce the power, could lead to results that are more driven by cell line than treatment. Now, the nice thing here is that treatment is balanced across the cell lines, so it shouldn't have too much of an impact on the results we see, but they could be improved by adding cell to our model. How we do that is when we first created the DESeq2 object, we gave it the design matrix, and we just said DEX in the first analysis we ran. And here we're going to say cell plus DEX, and that tells the function, okay, adjust for cell line, then test the treatment. So first, let's do some cell line adjustment, and then let's do a test of treatment. The p-values we're going to get back are still going to be testing for differences in treatments, but we're going to have this cell adjustment done and could make our results more powerful. Although the first time we ran this, we had a whole bunch of significant results, over 2,000 significant results. So not sure if that's really an issue for this particular analysis. It could be an issue for different analysis, for example, the one we'll look at in the lab section. The interpretation of the results is the same. The LFC for DEX just now represents the treatment effect after accounting for the cell differences. Similar to what we have in logistic regression and linear regression, those interpretations are after adjusting for here. In this case, it's going to be cell. How do we do this? Let's go through those. So we're going to the first model we did here. This is creating this in the same way that we have. We have the counts, we have the call data. Our design matrix is DEX. We add the referent level. Okay, we do the simple analysis and the simple results. And this is the same thing that we did before. I'm going to go over to R, and I'm going to run this code chunk. So first, it's doing the treatment only. You're going to see that run. In the second one, we are adding the cell line to the design matrix. So the calls are exactly the same, except for this has the cell line plus, and this one just has the DEX to it. We have the results for this. Compare the number of significant genes. So in the simple analysis, there are 2,700 significant genes. When we adjust for cell line, that number increases to 4,000. So adjusting for cell line, typically increases the number of significant genes because we've removed a source of unwanted variation from the residuals. The statistical gain from good experimental design and proper modeling. So the same principle applies here as in any regression analysis. Adjusted models almost always preferable when the design includes a blocking variable. For here, the design was clearly set up so that we have one cell line for treatment and untreatment. So clearly that was one of the things they took into account when they designed this experiment. So it's a good idea for us to account for that. So with this new cell model, we can again do all the same things that we did before. We can look at the top genes, and we can we could look at the MA plot. Actually, let's look at the volcano plot. I think the volcano plot is. Oh, we need these annotated results to do that. So let's get those first. Okay. Nope, that's not what we want here. It's the annotated results. Okay, I'm going to copy those down here. Still using the airway data. Nothing changes from that. We're just going to get the results cell here. And what we used initially was the results DF. So let's see where that was defined. All right, okay, that was defined right here. So I'm going to define that first. Give it a little different name from what we had before. So, so now I'm creating the results from the new analysis, turning into a data frame, adding the gene ID as a column, again, arranging by the adjusted p-value. We have the gene info up here. I think I did that wrong. Copy and paste it here. Good. Oh, that just did it for everything. I just going to call this res annotated cell. Okay, so creating a new object. Copy this, paste it in our studio, and boom, there's our top hits. Same top hit as we had previously. So the gene name that we had before, I think this is the same one. We can I can scroll back through these. Oops. Look at the previous plot. So that one again pops up as the most significant. P-value is lower than it was before. So it was times 10 to the negative 100. Now we're times 10 to the negative 130. And you see some others that have joined it up there. Number 2 is this CACNB2. I don't see that on the previous one. So that's certainly one that jumped up to be a lot more significant after we adjusted for the cell line. Oh, here it is right here. So it was relatively significant the first time, had a decent log2 fold change, but significance jumped way up to where it's around the same line that we had previously. And we could yeah, Malin, good question. So should we is using cell plus DX the same thing as DX plus cell? No, it is not. The ordering here is important. The last variable in the design matrix is the one that the DESeq is going to give us the p-value for. So this p-value that we get is still is there a difference between treated and untreated? Now, why did it do treated and untreated and not cell 1 versus cell 2 versus cell 3 versus cell 4? It's giving us treated and untreated because we listed DEX last. So the ordering here is important. And we want to keep the treatment variable last. Good question. All right, I wanted to look at the volcano plot. So here I'm going to look at that I'll just copy the whole thing. Let's take a look at it for the cell version. I don't think I need to change anything else. Oh, there we go. Okay, so here is the results from our cell-adjusted model. A lot more genes here, certainly. There's a lot more significance. And the upper end here, the significance here is a lot larger than we had before. So in this case, adjusting for cell line was certainly advantageous to our model. Oh, neat stuff. Okay, so let's wrap all this up, talk about, review everything we talked about this morning. So what we're going to do here, or what we did, was we build a DESeq data set. It's going to have a count matrix, sample metadata, and the design formula. After that, we filter low count genes, set the reference level. Then we run DESeq. In one call, it normalizes because it accounts for the fact that the sequencing depth is not the same among all samples. It's going to do the dispersion estimate. So again, that is where we estimated the dispersion, and then we had a little corrective factor here to increase the stability of those estimates. Statistically, that's my favorite part of this model. And then it does the model fitting. Once we have the dispersion estimate, we can do the model fitting. That gives you the p-value. So Benjamini-Hochberg procedure on the p-value, which gives us this adjusted p-value. We get the log 2 fold change. Positive means it's upregulated. Negative means it's downregulated. We can visualize these: MA plot, volcano plot, a heat map of the top genes. I think these all have different uses. Volcano plot, I think, is one that's really useful if we want to identify some genes for following up, because it shows us both the effect size, the log 2 fold change, and it shows us significance. So we want to see things in this top right and top left corner. Significance is important, but also the fold change is important. So again, something like this ZBTB16, this may be a little more of interest than some of these variables down here, which have a lot higher significance, but not as much, not as big a effect size to them. If the experiment has blocking variables or covariates, cell line, batch, sex, etc., let's include those in the design formula. That can help the power, give us a better analysis. So we'll talk about this more in the afternoon. We'll explore this for the Priscilla dataset, and tomorrow we'll return to this. We'll talk about some of the details of how this DESeq procedure works. We will talk about the false discovery rate, get into more detail about how that works to get the adjusted p-values, and we'll talk about clustering. Clustering is something we used for that heat map, so this will help us understand what that heat map is showing us. But clustering is also just a really useful procedure for you as an analyst to consider. Okay, so that's what I have for the morning. Is there any questions before we break for lunch? Great. Well, thank you for, I'll tell you, these first 2 days, this has been a real learning experience for me. As I said, I'm not a bioinformatician, so I was learning a lot about this stuff with you guys. So it was fun to learn about some of these procedures. What we'll talk about in the next few days is much more in my wheelhouse as a statistician, but this stuff was a lot of fun, and I'm looking forward to getting into the afternoon and doing some analysis there. So have a good lunch. I will see you back here at 1 p.m. and we'll talk more about these analysis for the Priscilla data set. I was going to give another couple minutes to let people trickle in here. All right, welcome back to the afternoon session here of our day 12 lab. Here we are going to be working again with the Priscilla database, Priscilla study that we began talking about yesterday in the lab. We saw the data set, we saw what it looks like. Today we're going to go on and do some analysis of it to get some of the differentially expressed genes. You'll have this loaded up, set up, okay, similar from yesterday. Loading the data in, similar to yesterday, identical column names and row names, all good. Okay, we have the study design here. Okay, this is where I want to start with this right here. Why do I have this here? Okay, well, let's go over to R, where I have the lab opened up. And I'm just going to run these lines here. I'm not going to run this whole parts. And usually, we get all these things and we kind of ignore what's on here. And this is kind of where sometimes things can go. So let's go back up to the top. So first, we loaded in tidyverse. We had some conflicts. So the filter function in tidyverse was masking the filter function in stats, and same thing with lag. Those are not big deals. Summarized experiment is going to mask a number of functions from the Dippler package. First off, count. Count is something that you may want to use in your analysis. For example, when you're counting how many are over or under expressed among the significant genes. What are some other ones that we will really care about? There's one I'm looking for here in particular. Here's the other one. Select. Select in the Dippler package is the most masked function out of anything in all of R. This happens all the time. There's another package called mass, M-A-S-S, all caps, very common package to get loaded in when it's like a companion package. So it's a required package for a lot of other ones, the mass is. And mass has a function called select. Dippler has a function called select. I cannot tell you how many times this trips me up. I'm trying to use the mass function. I'm trying to use I'm trying to use select, and I'm getting an error, trying to use select, getting an error, and I can't figure out what's going on. Well, what happens is R is trying to use a different version of select by default instead of the Dippler version of select. The select function we use a lot when we want to select, trim our data set down. It's one I use very frequently. So this happens all the time. So we can hit question mark, and you can see there's actually three versions of the select function. One in the Dippler package, of course, one in Biomart, one in annotation DBI. So these other select functions, they're totally different than the version that we really want to use. So I don't know why. It's just a very common name for a function, but I almost always want to use the Dippler version, so this trips me up a lot. Another one is filter. There are quite a few functions that will have a function called filter. And how this works is whatever library, whatever package you load last, that will be the package that takes precedence. So here, because we have this Pasilla package that, for example, loaded this annotation DBI, which had a select function, because we loaded that after we loaded tidyverse, okay, that's the version of select that R is going to use by default. So when I just type select, you can see it's going to use the annotation DBI package, that version of select, because that was the last package that was loaded in. It was loaded in when we loaded in Pasilla. So something that is annoying to say the least. So these three lines are stuff that I'm adding, and these tell R that, by default for select, use the Dippler version of select. So I do that by saying Dippler, double colon, and then select. Filter, double colon, filter, set it as the Dippler double colon filter, and count, set it as the Dippler double colon count. So by default, it'll use those versions. When you have a situation like this, another thing you could do, I could just use this every time I call the select function. I could use Dippler double colon select. So that tells R specifically, okay, use the Dippler package version of select. That's how you would tell R to do that. So annoying, believe me, this thing has tripped me up so many times, but there's not much you can do about it. So wanted to note why this is here, because in this version of the Markdown, I did not set those, and what you can do to just to make this a little easier, because to me, I always use the Dippler versions of these functions. Anyway, didn't mean to go off on a rant there about the select function. Let's move on here. We had the Priscilla data. Here's the metadata, study design data. We have condition, treated, untreated, 4 in the untreated, 3 in the treated. And then we had type, single read, or paired end, single read, or paired end. Recall from the lab we did yesterday, the principal components analysis, we could see both of these things having a notable impact on our results. So what we would want to do in this situation is when we run the DESeq function, we would want to have both type and condition in our model. We have type first because What we are interested in is condition, which we should have second. So whatever variable you're interested in, you put that second. If you wanted to see what genes are different by type, we would flip those around. But here we're interested in condition, which is those that are untreated and those that are treated by the Pacilla knockdown. Here we are setting the reference levels, filtering, so only keeping those that have at least 10 counts. After filtering, we end up with about 10,000 genes. Here we are running DESeq2, getting the results. And here we have the summary of that. We can see results there, and then the MA plot, which we looked at in the morning. The exercises, you're going to do 1 through 4, which are going to repeat what we did in the morning for this new analysis that we looked at. So getting the results in a nice format. How many rows are there? What does each row represent? How many genes have a significant result at 0.05? How many have a significant result at 0.01? Can use the same things we did before. And then of the significant results, how many are upregulated? How many are downregulated? What is the gene with the smallest p-value? What is the log twofold change for that gene, upregulated or downregulated? And print the top 10 most significant genes by p-adjust. Just repeating the things that we worked on in the morning. Size factors, getting those, and the dispersion estimates, getting the plot of those, and just, hey, what do we see from that? Next, compare the per gene dispersion estimates with the shrunken estimates for 3 genes. The gene with the highest mean expression, the gene with the lowest non-zero mean expression, and the most significant DE gene. So you can extract the dispersion estimates from these, and we can get the mean expression from this right here. And I may add some code in the chat to help you out with this one. Volcano plot, looking at that. Okay, what do we notice from those? How many significant genes are, plus minus outside of those thresholds and significant? Describe what genes are in the top right corner have in common. What about the genes in the bottom center? Just some information about those. And then lastly, to do a heat map using the functions that we did in the morning, and taking a look and seeing what you see, if there's any relationship with the type of sample that was ran. So do your best on these. I realize this is more questions than we've had in the past. I'll try to give you a little bit of help, particularly on this 2C. So I'll give you some code to help you out with that. Looking it over, it is a little more challenging, but everything else is really just following the steps and code that we used in the morning.